Hello grade 12s and welcome to Life Science, and today we're going to have a look at paper 2 revision. Right, so we've had a look at paper 1 and now paper 2's got a couple of things that we need to have a look and go over with. Okay, first one is DNA coding of life. That's the first one. We have a look at meiosis, and then lastly, genetic inheritance. Right, so what I'm going to do, I'll give you a couple of questions, and as I've given you the questions and I'm going to give you the answers, I'm going to explain how the answers come and how you know what to put there. Right, so here we go. I hope you got your thinking caps on, so let's have a look. First thing, remember I've always told you definitions, very, very important. So firstly, give the correct biological terms for each of the following descriptions. Okay, so that's what we're going to do. Let me see if I can fit them all on. I can. So let's see. A section of DNA molecule that codes for a specific characteristic. An allele that does influence the phenotype when found in heterozygous conditions. Next one, an allele that is not shown in a phenotype when found in a heterozygous condition. And then an amino acid chain consists of fewer than 50 amino acids. And then lastly, the production of genetic engineering copy of an organism using biotechnology. So they're using these big words, and make sure that you can take it down to its basis and explain what they are, right? There's 5 there, so I'm going to give you 3 minutes. Please make sure that you take your time and you concentrate while doing it. Are you ready? Off you go. Well, let's see how you concentrated. Right, so first one, oh, don't go so fast. First one, a section of a DNA molecule that codes for a specific characteristic. And let's have a look at that. That's called a gene. And when they're talking about a specific characteristic, like, for example, eye color, hair color. So that's when they talk about a specific characteristic. Okay, now the next one we have a look at here is an allele that does not influence the phenotype when found in the heterozygous condition. Okay, so it does affect the phenotype. Let's have a look at an allele that does, that does influence the phenotype when found in the heterozygous. And if we have a look at which one does, it can only be, come on, touch, it can only be your dominant gene. Okay, remember your dominant gene, if you have a heterozygous and you have a dominant and a recessive, the one is going to show up where the other one is not. Okay, and then the next one was exactly the opposite. It said there the allele that is not shown in the phenotype when found in a heterozygous condition. Of course, that one is your recessive gene, just your recessive chain, the one that is hidden behind everything else. Okay, your next one we're having a look at, an amino acid chain consists of fewer than 50 amino acids. What is it? Okay, now there's two ways of looking at it. I've put down a protein, just simple, okay? It's a protein. Now, what we can have a look at, it can be divided. It's most probably a quarterly protein structure, which means it's gone into 4 different stages, okay? But if I have a look at it, it's actually called a polypeptide protein. Poly meaning many, peptide bonds joining all amino acids. That will give you a polypeptide chain. Okay, so a protein is fine at the moment. Our next one that we had a look at is the production of a genetically identical copy. Oh, that's a big one. Identical copy of an organism using biotechnology. Okay, now this is where you come in with that funny thing, and they have done it. It's called Dolly the sheep. Now, Dolly the sheep was a clone. There it is, it's cloning. It was a clone. They made it where there's a little black dot 5 millimeters to the right-hand side of the left leg. It was the same with the cloned sheep. They are identical. Okay, so cloning we use during biotechnology, very important, and it has to be genetically identical. Otherwise, it's not cloning. Right, good, as long as hope you guys got those right. I know you did. So, very simple. Now, the last time we had a look at this section, I said to you, it's no such thing as match column A with column B anymore, okay? Now, each line has got a specific, you either have to pick A, B, both of them, or none of them. So let's see, what do they normally say? It says, only pick A, only pick B, pick both A and B, none. Those are your options that you have. Okay, so let's have a look at it. You have, I'm going to give you column A and column B. Let's see, we can go a little bit lower. There we go. I'm going to leave it up to there. I'm going to give you 3 minutes again. And with that 3 minutes, when it comes to 1.5, I'll just move it slightly down. I'll get rid of 1 and 2, and I'll bring in 4 and 5. So let's have a look at it. We have, they work made us aware of how genes are inherited. Okay, we're either looking at Darwin or Mendel. Okay, next one, a number of chromosomes in a normal human body cell. It's either 23 chromosomes or 46 chromosomes. The next one, the process by which DNA makes a copy of itself. It's either point mutation or shift mutation. Next one, a specific pattern of bands that represent a unique sequence of nucleotides that reassemble barcode. Okay, it's either DNA profiling or DNA replication. And then lastly, the structure that connects sister chromatids. It's either centromeres and centrioles, one of them. Right, so here we go. Remember, I'm going to give you 3 minutes. I'm going to let you go through 1 to 3. And once I've got to 1.5 minutes, I'm going to take 2 and 3, 1 and 2 away, and I'm going to give it the other 2. Right. So, remember, work wisely and think. Are you ready? Off you go. Well, these were quite easy, definitely. Let's have a look how you did, right? Remember, you actually got to put 2 and 2 together when you're doing these. These are very important. They do this because it can become confusing. Okay, so let's have a look. Let's change pens to get a good thing. It says, their work made us aware of how genes are inherited. Now, first one, Darwin, this guy here. Remember, Darwin has natural selection and all of that, right? So he says the strongest genes survive. So yes, he's bringing in genes moving across. But if we have a look at it, and we've got to look at it carefully, okay, you've got Mendel's laws, okay? And Mendel's laws, remember he brought in those peas. Can you remember the peas? And the long pea plants and the short pea plants. So when it comes to this question, I would definitely go with B, definitely, okay? The next one, this is quite a cool one. The number of chromosomes in a normal human body cell. Okay, now there's 23 and 46. Now, if I said to you in a sex cell, you could choose 23. Every other cell in your body has got 46 chromosomes. But then some of you are saying, but you got 23 pairs. Yeah, 23 pairs. This says 23 chromosomes. Don't make that mistake. Right? There are 46 chromosomes, 23 pairs in a normal body cell. So it is B again. It's quite easy, that one, huh? Okay, your next one, this is quite cool. The process by which DNA makes copies of itself. We have point mutation or we have frame shift mutation. Now let's have a look at it. Point mutation is a mutation that goes wrong at a specific point on your DNA strand. So let's say I have a triplet of CCC. Okay, now if I'm making a copy and I say, instead of going CCC, I copied it CAC, there is a specific point where it went wrong. Okay, and that is a point mutation. When it comes to frame shift mutation, okay, so if you have a look at me and they all sit like this, okay, what actually happens is when it's frame shift, by accident, everything shifts over slightly. So can you see there's one wrong there and one wrong there? That is frame shift. So the whole protein has changed. Okay, so that's frame shift. And here it is, the process by which DNA makes a copy of itself, it is none. Because they're both talking about reproducing or remaking it, but with a mistake. Okay. The next one we have a specific pattern of bands that represent a unique sequence of nucleotides that reassemble barcodes. Okay. Now, if I have a look at it, DNA replication doesn't do that. DNA replication is what happens when we're having a look at protein synthesis. Okay. DNA fingerprinting, that's where you get those funny lines, and you'll see those lines look, you'll see something that's got a thin line and a thick line like this, and then two very quick lines and a nice two long thick line like this. Okay, it's a DNA fingerprint. Okay, and that is how the DNA actually separates and sequences itself into a specific order. Right, so this one is definitely A. And then the last one, the structure that connects sister chromatids together, the way I remember it, it's very simple. Centrioles, poles. Okay, centrioles, poles. And centromere is in the middle, M for middle. Right, centriole is poles, centromere is in the middle. So centromere is the answer over here. So it's answer number A. Right, hopefully you guys are getting this right. Well, you know what, I know you are, so I'm not that stressed, right? You need a break, I need a break. I'll see you straight afterwards. Cheers. Welcome back. Let's have a look. I've got some new things for you, new questions. Now, let's have a look at this. It just says here, the diagram below represents a part of protein synthesis. I'm not giving it away. So you got to concentrate and you got to have a look. Okay, here we go. It says two questions. Well, three actually. Let me see if I can bring it down a little bit lower. Let's see, there's three questions. That's it. Okay, so I'm going to take it up to there. You can fit it all in. Okay, it is identify molecule labeled Y and Z. Okay, so Y and Z you got to identify. Name the phase of protein synthesis represented in the diagram. And then lastly, I'm going to move it up so that I can get to it. Give the name of the group of three bases that are indicated by number 4 on the diagram. Ooh, that's quite confusing. So what I'm going to do is I'm going to move this up again so that you can see everything. Hopefully it comes along. There we go. I'm going to give you 2 minutes to give this a shot, right? And it's actually quite easy. Let's see if you can get it right. Are you ready? Off you go. Let's have a look how you did. Now, these are not as difficult as you think. Okay, first question. Identify molecule Y or label Y and Z. So there's Z and there's Y. Okay, very important to have a look at it. First of all, Y is an anticodon. This whole thing, or should I say that thing there, is an anticodon as well. Okay, it's an anticodon and it's also called tRNA. So it's both. It's an anticodon because it's got 3, okay, and it's a tRNA molecule. And of course, Z is this whole strand from there to there, and that whole strand, it's very simple. That is a mRNA or a messenger RNA. Very easy. How did I know that that wasn't a DNA? How did I know it wasn't a DNA? First of all, I have a look at this guy here, at that letter U. There are no U's in DNA. There are only T's. And wherever there's a T, when it comes to RNA, it becomes a U. Okay, so that's the quickest way to know it. And then, of course, what gives off, you can see it separates after it's made, or dropped off its amino acid. Therefore, it's a tRNA, and this whole process is something else. I'm sure they asked that, but I'll wait for it. Okay. Next question. Name the phase of protein synthesis represented in the diagram. And it is translation. Remember the first stage where we make the mRNA from the DNA molecule. That is called transcription. Okay, so you're writing down the code. The making of the mRNA is actually writing the code down from the DNA molecule. Okay, and this part is rewriting it in a different language, in amino acid language. Okay, and that there is translation. Okay, it's quite easy. Next one. Give the name of the group of 3 bases that are indicated by number 4 on the diagram above. So what is those 3 bases together called? If this one is the codon, this one must be an anti. If that one's the anticodon, this one is the codon. Okay. Remember, it starts off with a DNA molecule has got triplets, 3 triplets, right? Then your mRNA joins those triplets to create a codon. That codon comes down and goes to the mitochondria, goes to the ribosome, and from the ribosome you get anticodons joining them, the opposite of codons. So the opposite of a codon is a anticodon. And what is similar to the anticodon, the code roundabout, will be your transfer RNA. Okay. Now, those were quite basic. I know you got those and you got those quite quickly. Let's have a look at the next question. I had a couple for you here. Okay, write down the base code from left to right that could be found at point 3 on the diagram. So you need to tell me what is the base code. And I'm going to give you a minute. This one's quickly. This is very quick. A quick minute. Are you ready? Off you go. Okay, let's have a look at it. Remember, it works the opposite, okay? So what goes with cytosine? Guanine goes with cytosine. What goes with uracil? Adenine goes with uracil, and adenine goes with uracil. So the 3 base pairs are very simple. It's GAA. There's your answer, GAA. Easy, simple. You can't go wrong with that one. Right, which is one that you got, and it's quite easy. Next one that we're going to have a look at, okay, still the same thing. It says here, the table below shows the DNA base triplets that code for different amino acids. So there it is. All I want you to do now is have a look at this. This is very simple. They're saying if you have TTT, you have alanine, okay? And, okay, sorry, lysosine. If you got alanine, you got CGA or GCG, any one of those. So the question that I'm going to give you here is write down the name of the amino acid represented by 1 and 5. So name number 1 and 5. So let's see. 1 and 5. So let's see. Make 100% sure it's at 1 and 5. 1 and 5. There's 1. So we're having a look at that one. And 5. We're having a look at that one. Okay. So what I'm going to do is see if I can do this. I'm going to have to go up to there. Okay, I'm going to move it. I'm going to give you a minute and I'm going to move it up and down slowly. So make sure you concentrate and know which one goes where. Okay, so let's start off with 1 minute and concentrate and do it properly. Are you ready? Off you go. Okay, hopefully you got it right. Now, first thing I would do is find number 1. And number 1 is very simple. It's ACC. ACC. And number Z will be CAC. So ACC, let's find ACC. ACC, there it is. So there's number 1. Okay, so number 1 is 309. Okay, and number Z is CAC. Let's see, CAC, can we find the CAC? CAC, I'm finding, come on. CCA is there. They can't give you one. Can you believe it? Number 5, we can't give it. Because look, it's CAC, opposite of G. Opposite of U, opposite of G. And if we have a look at it, there isn't any there. It's supposed to be CAC, and there's no CAC. There's CCA, so none of them are there. So you would say number 1 is, let me just make this a little bit longer. Number 1 is 309, and number 2 not available. Okay, number 2 not available because there's nothing you can do. Let's have a look at the next one. Okay, this is cool. This is where you need to look at. I'm going to read through it with you and make sure you concentrate. Okay, here we go. Sickle cell anemia is a genetic disorder which causes red blood corpuscles to collapse and form sickle cell shape. This reduces their ability to carry oxygen around the body. It causes by mutation of the DNA that codes for the protein hemoglobin. Part of the normal sequence of hemoglobin and mutated part of the DNA is shown below. So what we've got here is the normal sequence and the sequence with mutation. That's the sickle cell there, and that's the normal one. So let's have a look. Okay, it's quick 3 questions. This should take you quick. This is, how do we know that the base triplet provides on the table are those of DNA? Very easy. Name the type of mutation that occurs with sickle cell anemia, and explain how mutations affect the formation of a required protein. And I'm going to give you, with these, it's quick. I'm going to give you a minute. Are you ready? Off you go. Okay, so let's have a look how you did here. First of all, if we have a look, the first answer, these are quite simple. The first one, if you have a look at, they are all, come on, they are all T's. They've all got thymine. Okay, and a thymine is only on DNA, not on RNA. Don't forget that. Next one, okay, name the type of mutation. Now, sickle cell mutation happens at a specific point. And on that specific point, you're going to get, anytime. Okay, let's see if we can get this right today. Okay, it is point mutation. Okay, so your point mutation is your sickle cell, and then of course explain how the mutation affects the formation of required proteins. You are going to get a shift, a shape of the protein is changed. If you change the shape of the protein, you have a problem, right? Because remember, the shape of a protein is very, very, very important to how we're going to do things. Right, so when we come back, we're going to have a look at in detail and in-depth things, right? Let's see how good you are. So bring your thinking caps back, and I'll see you straight after the break. Cheers. Welcome back. Let's have a look at how new questions that I've got. Now, I'm going to start going into my favorite subjects, genetics. Okay, so let's have a look. You must concentrate, and let's see if you can get these right. Okay, first of all, okay, here we go. A guinea pig black fur is due to dominant B, okay, and white fur is recessive, small b, okay. Short fur is due to dominant big T, and of course, long fur is recessive, small t. Okay, a breeder did the following crosses. There's a cross: big B, big T, small b, small b, small t. And he says, using the genetic crosses, okay, determine the phenotype of the parents, which is quite easy to find out, okay, and the possible genotype of the offspring. Okay, so let's have a look at this. These are quite simple. Okay, remember, black fur is dominant. Okay, so we have big B, big T, small t. Okay, and we have them crossed with small b, small t. We know for a fact then that this is its genotype. I'm going to initial now, so that make sure you concentrate. And this is its phenotype. Okay, phenotype, it has capital, and if it's got capital, you're only going to see the capital, and the capital is the dominant, which means it's got black, and it's got short fur, if I'm not mistaken. Big T is dominant, and long hair fur, short, black and short, and this is white. I'm going to cross with a white and long fur. Okay, so that's a phenotype of the parents of the P1 generation. Okay, so we've got that down to a T now. Now what I want to do is I want to get us some space here. Let's have a look what we can do. Let's get some space going here. Now, they want to know what the genotype is. Okay, so we've already got the genotypes over there. They want to know what the cross is like. So let's get the crosses all together. What happens is you get meiosis, okay, meiosis. They're going to form the gametes, okay, and the gametes in this case is big B, big T, big B, small t, small b, big T, small b, small t. Don't forget your and signs. Okay, let me spread that out a bit, make it easier to understand. Okay, big B, big T, and big B, small t, and small b, big T, and small b, small t. There's the one, okay, and it's going to cross with the other one, and if we have a look, they're exactly the same. So it's small b, small t, and small b, small t, and small b, small t, and small b, small t. They're all the same. So I don't have to do too much work with this one. I know that it's all going to be the same. It's identical. Right, so we've got big B, big T. We've got big B, small t. We've got small b, big T. And we've got small b, small t. And all of those are going to be crossed with just one. Small b, small t. Don't have to cross it with too many. You're supposed to cross it with all of them, so maybe you should. Small b, small t, small b, small t, small b, small t. So all of those have to be crossed. Okay, let's bring this down, cross it there, bring that down. And you'll notice, make sure that you do these with a ruler, hey? You don't want to get confused. And we're going to end off with big B, small b, big T, small t, big B, small b, big T, small t, small t. I've got to make sure that you know that small t, otherwise we're in trouble. It's a big B and a small b. There we go. Small b, small t, small t. Okay, here we go. Small b, big T, small t. Small b, small t, small t. And there we go. All of them are going to be identical. All of them are going to be identical. Which means if we have a look at it, let me see if I can get more of this page going. There we go. Okay, we're going to have 4 of those. So we're going to have 4 big B, big T, small t, to 4 big B, small t, to 4 small b, big T, small t, to 4 small b, small t. So that is our whole genotype ratio. So let's see what they said here. They said, give the phenotype ratio of the offspring. Phenotype, okay? Phenotype, that is your genotype, okay? So let's get your phenotype going here, and we have a look at it. There's a capital and there's a capital. So it's black, and if I'm not mistaken, it's short. Okay, and there's a black and short, a black and tall, a white and tall, a white and short. So it's 1 to 1 to 1. So it's 1 black short, 1 black long, 1 white short, to 1 white long. Now, how can I say 1 to 1 to 1? Think about it. There's 4 of those for 4 of those for 4 of those. So for every one of these, there's 1 of these, 1 of these, and 1 of these. Okay, so the ratio is 1 to 1 to 1 to 1. It's that simple. It's not a difficult one. This was quite a cool one, even though you thought it was going to be difficult. Right, that's quite a cool one. I went through this one very, very quickly with you. Okay, now what I am going to do is I'm going to, I made sure that it was there. Should have written it there. What I am going to do is I'm going to give you this one to do. I did the first one just to do a recap for you because it was quite a difficult one. I'm going to give you 3 minutes to go through this. Okay? Concentrate and see if you can get it right. Are you ready? Off you go. Okay, let's see how you did with this. Okay, very simple. This is quite a cool one and a simple one. Okay, it's not a dihybrid cross. Dihybrid cross. It's a monohybrid cross. Okay, simple. We have an X and a Y, which is male, right? And we have a female, which is XX. Okay, and they said that the carrier of the gene for eye color is carried on the X chromosome. So we're having a look at the X chromosome. Okay, now we have genotype, we have a male, let's have a look, crosses, let's see, we have allele, here we go, yeah, a white-eyed male is crossed with a heterozygous red-eyed female. So male, white, crossed with a female, red. Okay, that's the phenotype, and the phenotype. Sorry, not genotype. That's your phenotype. And your genotype is, let's see, X, Y, crossed with a X, and it was a heterozygous, big R, X, small r. There we go. Okay, so now we're going to do the gametes. So remember, meiosis, right? And then it creates gametes. And the gametes are X and Y crossed with X, big R, and X, small r. And you're going to find fertilization. Okay, you're going to have X and Y, X capital R, X small r, and your genotype and phenotype, you're going to have X big R with an X over there. Okay, remember small, if I can do that in actual fact, otherwise we're going to be in trouble. Make sure, no, that's there. There we go. And you're going to, it's a small r, you're going to have an X with a Y, you're going to have an X small r, and you're going to have an X small r, Y. So you're going to end up, quite simple, it wanted the phenotype, if I'm not mistaken. It said give the genetic cross, show the female, use a genetic cross to show the F1 offspring. Okay, so we have your genotype, you have a ratio of 1 XX capital R small r to 1 big X small r, big R with a Y. You have an X, two small r's, and you have an X, Y, small r. And if I have a look at the phenotype of it, I have a female with red eyes, red, okay. I have a male with red eyes, okay. I'm going to carry on here, hey. I have a female with white eyes, and I've got a male with white eyes. Right, so there we are. That was quite simple, hey? Now, these are the next things that you're going to have a look at, but we don't have time for them. What this is they're going to show you that X and Y, okay, and hemophilia and that. Please, guys, you've got to learn these things. They're very, very important, and they will confuse you now and then, right? But go through them. I'm sorry that we're out of time, but I'm sure we'll catch up soon and make sure that you understand these. Speak to your teacher if you don't know what they are, and make sure that you do understand it. And we'll see each other soon. Cheers. Bye-bye.